Hi, everybody. Nice to be here, and thanks for the invite. This is a long time coming, so it's really nice to be able to have this kind of group all together in one room to talk about something that's certainly near and dear to many of our hearts, certainly mine. Today, over the next 15 minutes or so, I'm going to talk to you a little bit about some practical aspects of feed management in a zebrafish facility. Very basic logistical stuff. Oftentimes, I'm asked, well, for those of you that don't know me, I run a very large zebrafish research program at Boston Children's, where we have one of the largest, most complex zebrafish research programs anywhere in the world. And so I've got lots of experience in screwing things up. And I have lots of experience in practical aspects of managing this kind of resource. And so oftentimes when we talk about standardization and definition and improving some of the management aspects of this particular species, I'm the guy that tells you the annoying things that you don't really think about, because those things are going to limit the application of anything that we come up with in this room. And so what I wanted to do is talk about 2 or 3 things that are a thorn in our side now from a management perspective as it relates to nutrition. And I think it's really relevant to the conversation that we're having here today. So, let me see here. There we go. Sorry. Okay. So we are in what I would say a transition as it relates to zebrafish feeding from the traditional to a more progressive outlook. So the traditional approach as it relates to nutrition and zebrafish and feeding zebrafish is all about performance. And by performance, I mean primarily growth and particularly reproductive function. These are, without, if you can't do those things in managing a fish facility of this nature, you're not going to do any science, right? And so, in particular, there's a really large focus on egg production, embryo production in zebrafish. And this is a historic fact. This has to do a lot with the fact that the model, as a biomedical model, the fish as a biomedical model, really is born in developmental biology. So everybody was making eggs and embryos so that they could study them, right? And so everything was about what diet made fish grow and produce lots of eggs for experiments. That was the focus, and still is the focus in many labs around the world. And it's never going to go away as long as we're using this fish as a model. And so, not really concerned about what's in the diet, not really concerned so much about defining ingredients, defining amounts, limiting any ingredients, any nutrients, just shotgun approach. It's the best way I can put it. Just whatever works, right? And so a lot of the early diets came out of the fish hobbyist realm. So zebrafish is a tropical aquarium fish, and so the traditional feeding paradigm was built on a platform of Artemia or brine shrimp and flake food, tropical flake food. And we'll talk about this a couple times over the course of these next few minutes, but that was sort of the traditional approach, and still is the traditional approach. Many people still use that as the platform, as a nutritional platform for zebrafish, tetramin flake and brine shrimp. We're transitioning now more and more rapidly to a more progressive approach about nutrition, which still we have a focus on performance. We have to have a focus on performance. But we're also thinking about what diet creates a nutrition or a healthy representative animal for use in a wide variety of different studies, right? So a fish that produces embryos is not necessarily, at a copious rate, is not necessarily a healthy representative animal. So this focus on overall health, and then the definition of what I call the nutritional landscape. This is an experimental variable. a huge experimental variable that we have overlooked for as long as we have been using the zebrafish as a model organism. And you want to know what's in the diet in terms of the ingredients, but also the quantity. This makes a huge difference, how much you're actually feeding the fish. Go back, if you don't believe me in terms of the lack of a definition on these things in the zebrafish field, go in the literature now and try to look at a material and method section and try to find out how much and what fish were fed for a particular set of experiments. It's not going to be easy. In fact, it's going to be very difficult. So we are now, there's a growing awareness that this is important, as evidenced by the fact that we're in this room together here today. And so we're moving in this direction. But even when we figure out what it is that these diets should actually look like, there are still going to be things that we need to, problems we're going to need to solve in terms of, again, leveraging, so being able to apply what we learn. So we know exactly what the fish need. It's still a whole other matter of how we get it into the fish in a practical way. And so that's really what I'm going to talk about. So a few years ago at Children's, we started this transition in terms of trying to get away from the black box of nutrition and starting to define what it is we were doing and trying to have a data-driven biological basis for our feeding practices. And so what we did was we published a study which was looked at a number of different things, but one was feed frequency. So how many times a day do you need to feed a zebrafish in order to drive certain basic performance parameters? In this case, it was growth and importantly reproduction. We also wanted to look at simplifying the diet and moving away from this shotgun approach and feeding one diet once the fish hit the adult stage that we could define, or define to a much greater degree than what we had done before, control it. Also, the quantity, right? So we wanted to move away from this shotgun approach where we were using a mixture of zooplankton and various sort of off-the-shelf fish feeds, and we moved towards a aquaculture diet, what I would say is a commercial feed that was designed to grow cold water marine fish, but it's a fairly refined diet. It's a spreading diet, which many of you may know as Gemma Micro. So we used this diet. We did feed frequency experiments. We found lots of important things out. Number one, we found that you could actually feed the fish as few times as once per day and still get the same kind of performance as you could get with 3 or 5 times a day, that same amount. We also found that you could move away, you could remove something like artemia from the diet. So I often call artemia sort of the third rail of the zebrafish feeding set of beliefs, if you will. Most people think if you take artemia out of the diet for zebrafish, everything's going to end, right? And so we showed that you didn't need artemia. You could go to one commercial feed as little as once a day and still get good performance. This sounds elementary to some of you that aren't zebrafish people, but no one had ever worked this out. We just followed what was done before. So the interesting thing is we did this study. This was published in 2011. In the discussion section, we noted some of the problems that may arise as a result of what we were going to do. So we were going to transition to this kind of feeding approach, but we acknowledge that there may be challenges. And what I'm going to explain to you or illustrate to you over the next couple of slides are 3 of the primary challenges that we identified early on and are now grappling with now. And they're all very straightforward, particularly the first 2. So challenge number 1, feed delivery. So anyone that's worked in the zebrafish facility of any sort, particularly as they scale up, realizes what a challenge feeding is. So a zebrafish facility is a research facility is very challenging in some ways, I think, a unique challenge for feed delivery. That's different than commercial aquaculture. That's different from most other lab animals, mammalian lab animals. We have, in some cases, hundreds or even thousands of tanks. And so this particular, this photo in the background is a room that has 8,000 tanks in it. 8,000 inputs, right? That in and of itself, so it's one of the benefits of the model, right? So it's we can fit 200,000 fish in a room of this size, but there are consequences to that. And one of them is that there are many thousands of inputs for feed delivery. And these entities are not all uniform. If they were, it would be a lot easier, but they're not, right? So you've got different life stages, you have different densities. So we have a target density of 10 fish per liter, but it's not absolute. So we have some tanks that have 1 or 2 fish per liter or less, and then we have some that are 15 or 20. And it depends on experiment, depends on life stage. And then the experimental state is also very different. So you may have fish that are being used just to propagate, they're just being grown to propagate a subsequent generation. So you may be making a transgenic or a knockout, and all you're doing is growing up that generation to breed them once and identify carriers, right? Or you may be growing fish that are long-term tumor models or stem cell transplant models or anything. As we become more and more complex with what we're doing with fish, the number of different experimental states that can exist within one facility change. And all of those fish, all of those experiments, actually there are different nutritional requirements for them. So you shouldn't be breeding fish the same way you feed fish with a high tumor burden, adult tumor burden. But it's easier said than done. So this is a big challenge. And there is, I'm just going to do it like this. So it wasn't just you and your technological, although one.
Right over there.
Oh.
Doesn't want to work still.
Use the computer.
Computer errors.
To push them on the
Computer
computer.
there. All right. So delivery is a big challenge, and over the last 5 or 6 years, in particular, as we have transitioned away from the traditional feeding approach, and even if you're still doing it, there are lots of solutions for delivering known quantities of feed to hundreds, thousands of tanks. They include the venerable squirt bottle or squeeze bottle, wash bottle. We have
More technical issues. I'm just very lucky.
So we have insect pesticide or sprayers that have been used to deliver Artemia. We have modified and stainless steel versions of effectively what were spice dispensers to use to deliver known quantities of a dry feed to a tank. And then all the way at the highest pinnacle of technological innovation, we have robots that will deliver feed to hundreds or thousands of tanks. And these are all sort of representations of the fact that we have a challenge in terms of delivery. And so any diet that we design has to be deliverable. And preferably it's one that is easier to deliver. So if we come up with a diet that needs to be fed 15 times a day, this is going to be a practical challenge that we're going to have to deal with.
Sorry, the video feed has stopped for some strange reason. He was very lucky.
It's all good.
So can you check the video on there? No, on the pad. Can you tap the middle camera?
Yeah, they can switch the audio, they just can't see the speaker anymore.
Oh, yeah.
How you doing? What's Artemia? Huh?
So when can I start again?
Oh, okay.
No,
Just tell me.
You look good on the screen.
Do I? You're very kind. I can't even look at it.
It's working.
Okay.
We good?
Yes.
All right.
Sharing now?
Yep.
All right. So delivery is one of the big challenges. Right. So when we transitioned away from a diet that included zooplankton and a mixture of other dry feeds, we went to a single dry feed. We had to come up with a way of actually delivering it to thousands of tanks in a reproducible way. Not simple. And we've spent enormous amounts of time and money actually trying to figure out what's the best way to deliver a diet. The second challenge, if I can advance the slide.
You're pushing the arrow on the computer?
Yes, I am.
I'm glad it didn't happen to me.
You broke it.
And we started out the day with technical difficulty, and now we.
All right, it's all good. As long as you guys don't mind, I don't mind. The second challenge is waste production. All right, so what I have here is a figure that shows you a generalized version of the classic commercially available zebrafish housing tank. And most of the major vendors, they differ in some respect or another, but they mostly utilize the same approach, which is driven by high gradient flow into the tank, where solid wastes that are produced in the tank are then sucked out or siphoned out the bottom of the tank and up behind a baffle and out into the overflow drain. This is the standard approach, whether you do it with a siphon, whether you do it with just a baffle like this. These systems are all designed to be self-cleaning in the sense that anything solid that's going into the tank that's free, it's up in the water column, will get sucked out pretty quickly. This is the way these systems are designed, and these systems are not changing anytime soon. This is a realization that I've come to over the last couple of years when I've actually asked people to start, housing vendors to start thinking about this issue. It's not easy because we've got hundreds of thousands of units in production right now. This isn't changing. So this is the way these systems are made. And it's a challenge. It's less of a challenge when you're feeding a traditional diet, which is low levels, low biomass of Artemia going in, or rotifers, and floating, quasi-floating tetramin. That's really what these things were designed to deal with. Self-cleaning and that low biomass, low solids producing. That dietary regime produces relatively little solid waste. If you move to an aquaculture diet, a commercial feed, a pelleted diet or a crumble diet, and you feed it by a percentage of body weight, 3, 4, 5% of body weight a day for an adult, this is a sludge producer. Sludge producer. And this is something that we knew going in when we were transitioning to this approach. We knew this was going to be a challenge, but we did not appreciate how much of a challenge it was going to be. So this is sort of the backside of one of the housing units that's out there on the market today. That's the back. We're looking at the back of a tank, and that green is the baffle, and those plaques, if you will, those are solids that are starting to accumulate behind that baffle. And the second that that starts to happen, and they're starting to accumulate, because what happens first is you get a biofilm that forms, and then the biofilm is sticky, and then starts to, in this very narrow gap of 1 millimeter or 2, or whatever it is, I don't know what the exact gap is, but it's very narrow, it's very easy to start sticking solids. And when that happens, solids begin to, the tanks don't work the way they should. And so what you end up getting is actually more solids going directly into the system. You also get a lot of this waste directly to the sump. This is Artemia going into a tray filter. This is Artemia fed at high amounts, but we're trying to actually limit what's going in the sump. It's not easy. You just can't turn a system off and feed it. Like when I started in Zebrafish 20 years ago, we actually had these homemade systems that you had timers on. And we would put the timers on for 20 minutes to be off feed Artemia. And we would reduce this, but now the systems aren't very compatible, are really not compatible with this at all, so you can't do it. So you end up flushing that down the drain. That's not cheap. That's very expensive, and also causes all sorts of problems. And if you've gone through the pain and suffering of culturing Artemia, decapsulating Artemia, and purchasing Artemia, when you see this, it's painful. It's also, like I said, a sludge producer. So these are just pictures of sumps in a particular, on the left it's a picture of a sump. That is sludge that's just accumulating. On the right is a tank that doesn't have any fish in it. So that's all the suspended solids in the water, and the fish keep up that suspended solids by design, get it up into the water column so it gets pulled out. But if the fish isn't there, anywhere where you have sort of settling zones like this, this is what you'll get in a fish system that's fed intensively with a commercial feed. Any commercial feed, doesn't matter which one it is. And that sludge is not a good thing. One, it promotes the growth of lots of things. So do you see, I don't have just have a laser? Doesn't really. At any rate, if you see the little lines on either in the sump and on the tank, those are bryozoa. That is a major problem. A following organism that acts as a scaffold for lots of other microorganisms. That could be problematic. It's also nasty. It's a major problem in the shipping industry. Not this particular species, but bryozoa is a major nuisance species, and it's a nuisance species in zebrafish facilities, particularly when there's lots of suspended solids in the system. That solid waste is a problem. I don't know if this video is going to work, probably because I'm a Mac person, it won't. Oh, it is. Look at that. So these are fish. They are between feedings, and they are grazing all along those solids. They're grazing on this stuff. So microorganisms that are in the sludge. So if you have sludge in your system and you want to have a fun little observational experiment, pull some of that sludge out, look at it under the microscope. It's going to be crawling with microorganisms. What the impact of these microorganisms is not necessarily known, but certainly we know that certain microorganisms are vectors for different pathogens. This is something you generally want to avoid, but we are producing systems that are chock full of it. The last challenge that I'll talk about is a little bit more complex, and it has to do with nutritional imbalance. So we transitioned from a zooplankton-based diet to one that is completely reliant on a commercial feed that was designed, again, to grow cold water fish. And it's designed to grow fish quickly. And it works really well for zebrafish. It's not nutritionally formulated for zebrafish. Obviously, we don't know what the nutritional requirements are. We obviously know that. So, but this diet and other diets work well to grow fish. And they grow zebrafish very, very well. And one of the things that you see is that when the fish are in rapid growth phase, so zebrafish, it displays sort of indeterminate growth, but the growth slows down. So if you leave a fish alone for a long period of time, it'll be gigantic in 3 years, but that growth slows. So they hit a growth plateau. So these diets, and this diet that we use, is very good to get the fish to that plateau. Very good. It's good at growing, driving somatic growth. But once they hit that plateau, where do all those excess nutrients go? So this is this feed that we use is 62% crude protein. which is a lot more than is required for an adult zebrafish, or even a growing zebrafish. This, in part, is because it's designed to mimic Artemia, which has that similar protein content, about the same crude protein content in Artemia. But the idea is, once they hit that plateau, what happens? Where does all that excess nutrient go? Right? We don't change what we feed. We don't change the formulation of the diet. We don't necessarily even change how much we feed, not deliberately. So we feed 1% a day, 5% of body weight, 4 to 5% of body weight, but we're not generally adjusting that for every single tank. And it's in a representation of thousands of different units. So what happens is you end up feeding them that same high protein, high nutrient load diet that they don't necessarily need. And so there are lots of repercussions to this that many people in this room can talk about with greater specificity than I can. But certainly one of the things that happens from a practical and logical standpoint, and particularly in females, is that once they are done growing, they are allocating those excess resources to reproduction, right? So female is making eggs. And so one of the things that we see is they become egg machines. But the question is what happens then? It becomes very difficult to balance things like viability and embryo production. So one thing that we've done over the years and we've seen happen is our viability rates go down. So the number of eggs that are good has decreased. So a viable egg is an embryo that on day one is normal and healthy. And so one of the things we look at is what's the relationship between that ration size and the breeding interval in an adult fish. So female is taking all that excess nutrient and using it to build and to produce eggs. You've got to take those eggs from her to sort of compensate. Do you understand what I'm saying? And so, but how many eggs do you have to take? What's that interspawning? What's that breeding interval look like? Is it every day? Is it every other day? Is it once a week? Is it every 2 weeks? And again, this is once the bulk of somatic growth is completed. So what you get are these great big females that produce lots and lots of eggs. So we did a very simple experiment. Actually, it wasn't very simple. It was actually enormously burdensome to do. But we tried to look at what the balance was in females. And I'm just telling you this to illustrate the difficulty of this one aspect without having a feed that's designed for the species in question and the life stage. So we fed fish either 1, 3, or 5% of body weight a day, once a day. And then we bred them either twice a week, once every other week, once a month, or once every 3 months. And we wanted to look and see what the relationship between embryo number produced and viability was. i.e., or to think about it, is that somewhere there's a sweet spot where you get the right amount of feed in the female such that you have a highest amount of embryo production possible with high viability. So there's no waste. Because what happens is they produce and produce and produce. They're ovulating all the time, and if you're not taking those embryos away, they expire. Essentially, within the body cavity, they expire, and when they are expressed during spawning, they don't fertilize. So that's the relationship that we were trying to look at. So this is just graphs that show the relationship between fecundity, which is the number of embryos produced, and viability. And you can see that the relationship is not necessarily, we couldn't figure out where that sweet spot was. So regardless of whether or not we bred them once, so A is twice a week, B is once a week, C is once every other week, and D is once a month, E is once every quarter, essentially. So we weren't seeing much of a difference with the interspawning interval, but we were seeing the difference with the number of embryos spawned overall. And conversely, we saw that we had higher viability when we fed them less. But we're still seeing viability rates which, so there's a balance. So you either don't feed them, if you feed them 1% of body weight a day, you get pretty low fecundity. If you want higher fecundity, you get much higher inviability rates. And somewhere in there is a sweet spot that we can't necessarily hit. The sweet spot would be that you would have higher fecundity, higher viability, so that you're not wasting time and energy. Because if you have high so you need lots of eggs, but they need to be good for you to be able to do these experiments. So just an illustration of how difficult it is actually to get it right. So in summary, what I would list as requirements for the next generation of zebrafish diets is that obviously they have to be comprised of defined ingredients that can be controlled. They should be able to produce and maintain representatively healthy fish by default. That should be the default diet. Whether we have maintenance diets, whether we have breeding diets, these are things that I think probably should be discussed. Delivery of whatever the feed is has to be practical. It's got to be, the feed itself has to be compatible with the current housing system such that its administration does not overtax it. Any pelletized diet is going to cause problems. So we're going to have to think about different ways to formulate a diet physically so that you don't have that wastage going into the system. But at the same time, you're going to have to be able to deliver it in a practical way. And it has to be life stage and experimental context specific. So there you have it. I'll take questions if you have them.
I have a question for Chris.
So, are you say you feed flake diet?
No, we don't.
You feed extruded diet?
Yes, it's an extruded pelletized diet.
So why standardize on an aquaculture pellet diet rather than on flakes like tetramin? If you want to standardize on something, it produces more sludge and is not optimally suited to zebrafish, as you showed.
Yeah, there are a lot there. That's a good question, and there are lots of different reasons as to why we moved away from flake diets. Number one, a flake diet to feed it properly is not easy. So a pellet, the difference between the physical characteristics of a pellet make it easy to deliver at least known quantities. A flake is much harder to deliver known quantity. Oftentimes, historically, people would take flake and they would mix it with artemia, right, and fluidize that. That, among other problems, would lead to leaching of all the water-soluble nutrients from the flake directly into the water without getting to the target fish. The other thing about flake diets is you have almost no control over what's in them, right? And so when there are people in this room who can talk at length about the problems with flake feeds for our research fish. Now, does it work in terms of supporting growth and reproduction? On its own, I would doubt it, but in conjunction with other things, yes. But we moved away from that for those reasons and others. Yes?
I would just throw out a comment that the switch to Gemma that's happened to Gemma microfood, it really came down to the fact that the food would stay viable and palatable for a couple hours at the bottom of the tank. And until that food came out, most of the time you'd never see fish eating from the bottom. But that pellet material, they just will eat it all over many hours, where the flake and the other stuff quickly just rotted and the fish could care less. So it was that feature, this is how scattering is cornering the market, if you will, and Chris's paper showing that you could just do this once a day. But it has a lot of problems, as he pointed out.
Okay, last question.
Is it me or him?
Ron, go ahead. You won.
I won, thanks. This is kind of a dumb question. I'm not a zebrafish guy, but do you know the point at which these fish begin to switch energy allocation from somatic growth to ovarian gonadal production? I know it's not an abrupt change, but there's a, by looking at, say, gene expression of myosin or some of the reproductive hormones.
improvement problem pretty easily.
I don't know that specifically what it is, what that landmark is. And obviously there are lots of things that would influence this, but it's typically when they are, well, there's a lot of spread in this, but it's at 3 to 6 months of age. When the fish has hit about males probably about 3.3 to 4.4 grams, females 0.5 to 0.8 grams. Once they hit that size, they're definitely allocating anything towards reproduction and very little bit to growth. The switch probably happens earlier than that. Steve probably has the best information on that, but it's very variable, and we don't have really any published metric for that. That's standardized.
There is, I can't remember the details, but there is a very good paper on this in PLOS One by Matthias Hammerschmidt's group that looks at basically allocation to energy being deposited in lipids, somatic growth, and egg production. And like I said, I can't remember the specifics, but we should definitely consider that paper in any further kind of discussion.
Yeah, did that come out like in the last year or two?
2016, I think. Yeah.
Okay, thanks.
And I think it's a lot earlier. I think it's between 1 month and 2 months of age that they start.
One more comment. We can talk about
Question,
it.
Chris. It has to do with, like the work that you do with frequency. Have you ever thought about the time of day of feeding as being something important, or is it just logistically prohibitive?
It's a good question. It's logistically prohibitive generally, but it's a good question that should probably be answered. I think fish are generally fed during the 9 to 5 workday. It's probably the best way to put it.
And again, there is a good paper on time, and I can't remember the specifics, but on when fish are fed with regard to how much fat they store in adipose tissue. So I can dig that out as well.
And in humans, ApoB, intestinal ApoB is on the clock.
Yeah, I mean, I think everything, well, a
Which
lot of
makes
things.
all the atherogenic lipoproteins is cycles.
And it's all time maybe save it for the break, I think, or for the panel discussion over it.